Welcome to this lecture on the basics of neuroscience. Today, we are going to explore one of the most fascinating processes in the human body, how brain cells communicate with each other. We will dive deep into the microscopic world of neurotransmitter receptors, specifically focusing on ionotropic and metabotropic receptors. By the end of this session, you will understand exactly how your brain processes lightning-fast reflexes and how it manages long-term changes like learning and mood. Let us begin our journey into the synaptic cleft. To start, imagine you are standing on one side of a river and you need to send an urgent message to a friend on the other side. You cannot shout across, so you put a written message in a bottle and throw it across the water. In the brain, neurons face a very similar problem. They are not physically connected. Instead, they are separated by a tiny fluid-filled gap called the synaptic cleft. To cross this gap, the first neuron releases chemical messengers called neurotransmitters. These chemicals act exactly like that message in the bottle, floating across the space to deliver critical information to the next cell. Now that we know how the message crosses the gap, we must ask how it is received. The second neuron has special receiving stations on its surface called receptors. You can think of a neurotransmitter as a highly specific key, and the receptor as a highly specific lock. Only the right key can fit into the lock to trigger an event. Without these specialized protein molecules embedded in the cell membrane, the chemical message would just float away, and the brain would be completely silent. Broadly speaking, the nervous system uses two main types of these locks to process information, which we will explore next. Building on our lock and key analogy, let us start with ionotropic receptors, which are truly the sprinters of the nervous system. Imagine a locked door with a built-in keyhole right on the handle. The moment you insert the key and turn it, the door swings open immediately, allowing people to rush inside. Ionotropic receptors work exactly like this direct door. Formerly known as ligand-gated ion channels, the receptor and the channel are the exact same molecule. When the neurotransmitter binds, the protein physically twists and opens a central pore. This allows ions to rush into or out of the cell, creating an incredibly fast electrical response in just 1 or 2 milliseconds. This speed is absolutely critical for rapid reflexes, like pulling your hand away from a hot stove. Since these ionotropic receptors open channels for ions, we can actually calculate the electrical current that flows through them using a variation of Ohm's law for cell membranes. The formula is I ion equals G ion times the difference between Vm and E ion. Let us break down these symbols. I ion represents the electrical current of the specific ion flowing through the channel, measured in amperes. The symbol G ion represents the conductance, which tells us how easily ions can pass through the open channel, measured in Siemens. Vm is the current membrane potential of the neuron, and E ion is the equilibrium potential for that specific ion, both in volts. The term in the parentheses is the electrochemical driving force. If this driving force increases, or if the conductance increases because more receptors open, the total ionic current will increase proportionally. Now that we have our formula, let us look at a practical example to see how fast ionotropic receptors change the electrical state of a neuron. Suppose glutamate binds to an AMPA receptor, opening a sodium channel. We are given a conductance of 20 picosiemens, a resting membrane potential of -70 millivolts, and a sodium equilibrium potential of +60 millivolts. First, we convert our millivolts to standard volts. Then we substitute these values into our equation. We calculate the driving force by subtracting 0.060 from negative 0.070, giving us an inward driving force of negative 0.130 volts. Multiplying this by the conductance gives our final answer. The final sodium current is negative 2.6 picoamperes. Physically, this means a tiny but rapid stream of positive charge flows into the neuron, which can help fire an action potential. Having seen the lightning-fast ionotropic receptors, let us now explore metabotropic receptors, which are the marathon runners and managers of the nervous system. Imagine you arrive at a secure building, but instead of a keyhole on the door, there is only a doorbell. When you press it, you do not open the door yourself. Instead, a security guard inside hears the bell, walks over to a control panel, and unlocks several doors at once. Metabotropic receptors work just like this doorbell. The neurotransmitter binds to the outside, but the receptor itself does not have a channel for ions. Instead, it sends a signal inside the cell to trigger a massive biochemical chain reaction. This process is slower, but it allows for incredible control over the cell's state. To understand how this doorbell mechanism actually works inside the cell, we need to look at the proteins involved. Metabotropic receptors are formally known as G-protein coupled receptors, or GPCRs. When a neurotransmitter binds to the outside, the receptor changes shape on the inside. This activates a special helper called a G-protein. The activated G-protein breaks away and activates other proteins called effectors, which then synthesize second messenger molecules. These second messengers float throughout the cell, altering metabolism and even turning genes on or off. Because this involves multiple steps, it takes anywhere from tens of milliseconds to several minutes. However, this widespread influence is crucial for neuromodulation, allowing your brain to control complex states like your mood, sleep cycles, and long-term memories. One of the greatest advantages of the metabotropic system is signal amplification, which we can model mathematically. The formula is n final equals n initial times a raised to the power of n. Let us define these terms. N final represents the final number of activated molecules, like second messengers. N initial is the starting number of neurotransmitter molecules binding to receptors. The variable A represents the amplification factor, meaning how many new molecules one single activated protein can turn on at each stage. Finally, the exponent n represents the number of steps in the biochemical cascade. As the number of steps or the amplification factor increases, the final cellular response grows exponentially. This explains how a tiny chemical signal can completely change the state of a neuron. Let us calculate how powerful a single metabotropic receptor can be using our amplification formula. Suppose a single molecule of norepinephrine binds to a receptor, so N initial is 1. This receptor activates a cascade with 3 distinct steps, activating G proteins, then adenylyl cyclase, and finally producing CAMP. So N equals 3. If the amplification factor A at each step is 10, how many final CAMP molecules are produced? We substitute our values into the equation, 1 times 10 raised to the power of 3. 10 to the third power is 1,000. Our final answer is 1,000 molecules of CAMP. Physically, this means just one neurotransmitter molecule altered the state of 1,000 molecules inside the cell, demonstrating the massive power of the doorbell mechanism. Now that we have calculated the effects of both systems, let us look at this diagram which perfectly summarizes their fundamental differences. On the left, you see the direct, fast action of the ionotropic receptor. Notice how the receptor and the ion channel are the exact same physical structure. The neurotransmitter binds, and the ions flow straight through the open pore, visualizing our key and lock analogy. On the right, you see the indirect action of the metabotropic receptor. There's no hole for the ions. Instead, the receptor uses a G protein middleman to activate an effector enzyme. This visualizes our doorbell analogy, showing how one signal on the outside creates a massive, amplified cascade of events on the inside. To wrap up our lecture, let us review the most important ideas we have covered today. First, ionotropic receptors are your brain's fast, direct, ligand-gated channels. They mediate rapid behaviors like reflexes and real-time sensory processing. Second, metabotropic receptors are the slower, indirect G protein-coupled receptors. Instead of opening immediately, they trigger biochemical cascades that allow for massive signal amplification, which is essential for long-term changes like learning and mood regulation. By utilizing both of these distinct systems, your nervous system achieves a perfect balance. It has the lightning-fast speed it needs to survive immediate dangers, and the slow, adaptable flexibility it needs to learn, grow, and feel emotions. Thank you for joining this session on neurotransmitter receptors.